tonight, I'm very pleased to introduce Justin Siegel. He's actually was an undergrad at Davis, and then he went up to Seattle afterwards, and then we were lucky enough to recruit him back down here. And he's actually has a weakness he won't tell you about, but his weakness is chocolate. He will do many things for chocolate.
Anything, really.
Anything. There you go.
I'm not embarrassed.
So anyway, without further ado, he's going to tell us about the interface of the really interesting work he's done with video games, crowdsourcing, and sort of getting science out to the general public. Justin.
Cool. All right. Well, thanks for having me here. Let's see. I was excited to be here to kind of talk about some of the things. I never really thought I'd be getting into video games and science. It just sort of happened, but it's really exciting to be able to come here and talk about it to such a broad community. Oftentimes when I give talks, they're on a very special subject that there's a specialized group of people that are interested in. I'm assuming everyone here likes video games. Yeah? Or I'll make that assumption at least, or at least knows what video games are. So I would say pretty conclusively at this point, video games have had a major impact on society, right? They've really go throughout all of culture at this point, and everyone has probably played a video game at some point or another, with the first major one being Pong back in the 70s when they first started up, even though some people say they started up in the 1940s. And since the 70s, they've really come a long, long ways, right? They've moved from a simple black and white, 2 bars moving up and down and hitting a little white ball back and forth, to very, very intricate games that they're either visually beautiful and have very intricate storylines that go on like over and over, where you have these massively multiplayer online games where thousands or tens of thousands of people are all playing together in a team-based effort to go and try to achieve some kind of goal, right? So that's great. That's hugely entertaining. That's a multi-billion-dollar industry at this point. But you could argue this doesn't really contribute back to society. And hopefully what we all want to do somewhere deep down inside is make the world a better place, right? And make it better for the next generation. And so it'd be nice if we were able to take these concepts and the interest and the excitement about video games and start putting it into a context that does actually do that and does make the world a better place. And so that's really what the talk today is about, is how do we go about doing that? And I would say it actually probably really started in grade schools. Are you guys familiar with Brain Pop? No? All right. Well, it's in about 25% of schools around the US at this point. And it's a hugely popular game for teaching basic science, math, English, health, and all that stuff. If you have a 2-year-old daughter like me, there's another game that's called ABC Mouse that you'll see advertisements for again and again and again. But they're starting to hit into education. So using video games to engage students to have them learn about stuff, right? And so that's excellent. That's kind of going from entertainment to education, but that still doesn't make that impact that we want, right? It's still not going all the way there. And so what I would say is entertainment is where they started, and clearly video games have dominated in that market. They've gone into education. Brain Pop's, like I said, in 25% of the schools around the country, which is pretty phenomenal. And that's a very fast-growing industry. So it's clearly in education at this point. But it can be used for scientific discovery, for moving past what we already know into the realm of unknown, like enabling people to skip the 20 or 30 years of education and going straight to the questions we want to address within a very short time frame and an easy and fun to do arena, I would say. So that's really where the whole talk's going to be focused around today. And so my specific field, is computational and molecular modeling. And so essentially what I do is I'm very interested at looking at protein structures, and I'm actually showing you what a protein looks like here as it folds up, and thinking about how it folds into a 3-dimensional shape. And when that protein folds into a 3-dimensional shape, it imparts function on that protein. And that function dictates essentially everything we do in the world around us. Nature uses proteins for the vast majority of things it carries out. It performs essentially all the chemical reactions in your body. It makes up your hair and practically every part of your body, as well as all of nature around us. So proteins are incredibly important, right? So we'd really be able to like to rationally manipulate them to better understand maybe new drug targets, or even rationally manipulate them to create new proteins to do new things that maybe nature didn't evolve for, but we really want as society. And so a lot of our research goes into designing and modeling these proteins. And so what I'm showing you here is, it started in 2005, it's called Rosetta at Home, and it was a screensaver. That's all, it's not really a video game yet, it's just like the first step towards a video game. It's just a screensaver. And people would essentially load up this screensaver when they went away and their screensaver turned on. Instead of just showing pretty pictures, it would start donating their computers to our lab, and we could run simulations on people's computers all around the world, right? And so that was pretty cool. So we have about 300,000 people at this point. They're donating their computers to our scientific research. And we were quite excited about this. This makes it so I don't have to spend hundreds of thousands of dollars on computers. I can just say, oh, please go download this software, and when you're not using your computer, I'll use your computer. And so, it definitely led to some advances. But what you'll notice here is this is just kind of going through and changing the structure of this protein. And people actually started looking at this and liking it, and they were saying, I see what this computer is doing, but it seems like it's doing funny stuff. Like you essentially want, and what I'm showing, you can't quite see it here, this right here is what the protein is supposed to look like. And you can see what it's sampling here doesn't look at all like that, right? And it takes a really long time because we go through a random process to try different conformations and different structures of this protein and then evaluate how likely that protein is to actually take that form. And the one with the lowest energy is often predicted to be the best one. And the hope is that the actual protein structure is the lowest in energy, or the lowest scoring, right? It's like golf, the lower score, the better. And so so in 2005, this Rosetta at Home came out, and this was from David Baker's lab at University of Washington, where I was doing my graduate work. And we got a lot of users, and we started getting questions from users saying, can I do something with this? This doesn't make sense. I'd like to actually be able to manipulate what's going on here instead of the computer just doing it. And we kind of looked at that and we were like, well, that's sort of interesting. We didn't even realize that people would be interested in that. But we had that request over and over and over and over again. And so we contacted a professor at the University of Washington, Zoran Popovic, who specializes in games and trying to figure out how to make human interactive software. And we're like, we have this software. It's folding proteins, which is important for society and to be able to do. And people want to help us. They actually don't want to just donate their computer time. They want to donate their actual time to carry out these folding simulations. And so he was like, well, that sounds pretty neat. Let's build some software to do that. And so that was what led to Foldit. And that's at the time, that was 2008. This is the first, as far as I'm pretty sure, I'm like 99% sure, this is the first at least majorly distributed interactive scientific game where hundreds of thousands of users have downloaded and played it. It's been recognized by President Obama with an award. It's been recognized by essentially every news article and mainstream media that you can imagine for all the different breakthroughs that have happened through it. And it's really just because a couple of people said we'd like to be able to try manipulating these things. And so what I'm showing you here is the actual interface to this video game, where it's showing you a protein structure. That's that green wiggly thing with all the stuff coming out of it. And you're seeing in real time the reds are bad interactions, and you can the players selected a region and allowed the simulations to say, well, hey, if you move that, that's better. Oh, that one's now better, and that one got better, and that one got better. And in real time, the score gets better as well. And so in real time, people can start manipulating the structures of these proteins and evaluating, is this a good interaction or a bad interaction? Is this likely to be what this protein looks like or unlikely to be what this protein looks like, based on the game and interacting with the game. So this was very exciting. This was in 2008, and we really started trying to understand is how, the people playing this game were guided by the score and their intuition of how shapes fit together, because that's all it is. It's like 3D Tetris. How do shapes fit together? And so we had the question of, how useful is this actually going to be? It's great that we made it, we send it out, and people are playing it, but does it actually lead to scientific discovery, right? And so that's essentially what I'll spend the rest of the talk going through, is giving you 3 different examples where I would say, yes, it does lead to scientific discovery. And so the first one was right after the game was developed. We wanted to know if people could actually engineer novel proteins that have never been seen before in nature, something that the players actually came up with, not something that they were just saying, this is what it's supposed to look like, can you get back to what it's supposed to look like, saying, what do you want it to look like? And then seeing if they can actually make something that works that does that. So this protein right here is a protein that I engineered as a graduate student. I spent about 2 years working on this protein to bind this molecule here, and the protein here is in green. But I've had, at this point, almost 20 years of experience in the field. I'd consider myself an expert. Yeah, I wouldn't say it's quite a fair comparison that I can use a video game and do that. That's not really saying that the general community can do this. But what I can tell you is I spent 2 years, I engineered this protein to carry out the function of interest, and that was the first 2 years, but then I spent another 2 years trying to make it better. And I could not make it better. I really did actually try very hard to change the structure around this molecule here to try to improve the interactions to make this protein carry out improved functions. Couldn't do it. I put it on the shelf. I said it was worth a shot. I tried my best. I'm not going to try it. Right as I was going to put it on the shelf, a game folder came up, right? And I was like, well, maybe I should send it out to the online community, because I tried all the ideas I had, but maybe someone else out there who hasn't kind of been thinking about this in the one way for a decade has a new idea or a different way of looking at it, right? As opposed to someone that's been trained down to have a particular vision of how proteins are supposed to look like. And so at the time, I also had an undergraduate who was a junior, or he was a sophomore at that time, actually. He was a sophomore at that time. He'd never taken a biochemistry course, so I had just gone to the lab and had just trained on how to do basic wet lab biology through a program I'll briefly mention later. And so I was like, okay, seems a little too high risk for me, but for an undergraduate, this sounds perfect. And so the idea, and he was thrilled about this, the idea was that he would act as the intermediary. He would take the protein, send it to the online community, the hundreds of thousands of users. They would mess around with the protein, and I'm going to go back a slide real quick. And when I say they would mess around with the protein, we essentially, what's in green here had to be fixed. They were not allowed to touch any part in green, because we knew that was the core of the protein. And we didn't want them to mess with the core. We just wanted them to add new interactions in this area. And so we allowed them to do whatever they want in the orange areas. They could break it open and add a bunch of stuff inside. Oops, wrong button. They could close it down. They could change the structure. But they literally could do anything they wanted to it. It was just a game, and they got a score back as they did whatever they want with it. So they would go do whatever they want with the protein. Chris would get back the solutions. Chris would go through the solutions and select ones that scored well, looked interesting, or were very structurally unique. And then he'd go into the lab and start actually testing them. He'd build those proteins and see if they did what the players wanted them to do. Clearly, Chris is not biased in what he's picking. I show him a picture of playing music here because he very explicitly said to me that he went to college because his father forced him to. He wanted to be a musician. And he was very bored and not very excited about college, but then the idea of this, of doing real live research, kind of came to him through several different people, and he was like, oh, maybe this will be interesting. But I can tell you he's actually quite successful now. He still plays music, but now he's at graduate school at Berkeley and doing very, very well. He definitely didn't come from the protein design background, to summarize it. So this is what they started with. I'm zooming into one part of the protein because amazingly, we sent that structure out. We let them do whatever they want all around the protein. All of the players focused on one specific loop, this one specific region of the protein. They didn't even, a couple of them messed with other places, but the vast majority of solutions that they sent back to us and submissions they sent back to us were all messing around with this region of the protein. So we were like, well, that seems like there's some kind of group mentality there, that this seems to be the best. And very consistently what they did is they turn this random loop that kind of sticks out into a what's called a helix turn helix, and you can kind of see it's helix turn helix. And instead of going out, it goes in, right? And now instead of having a big space here, it covers the ligand. Literally, the title of the puzzle that we sent out to them, cover the ligand. And I will say that all the players, before they play Fold It, and they go through a series of tutorials. And those tutorials teach them some of the jargon, like what a ligand is and what loops are and helices are. And so they know what that jargon is. I didn't introduce it to you too much because it's not the main point. I think everyone can see that there's a big hole here and now there's not. So I figure that's good enough. And so we were like, well, that's pretty neat. That's a very unique structure. I took out that structure and looked for similar structures that occurred in nature, and it does not occur in nature. I'm not going to say there's no helix turn helix motif, but in this exact orientation, there's nothing similar to it in nature. So no one just went out and looked what happens in nature and picked it out. They came up with it on their own. So we were like, well, that's neat. That's a very interesting structure. Chris started testing it. We ended up working with collaborators and got an actual structure of this protein to see exactly what it looks like. And what I'm showing you here is what the players designed, and I always have to look at this. The players designed the purple here, and the crystal structure, the actually experimentally determined structure of this protein, is orange. That's pretty phenomenally similar. If you're in this field, you would be blown away if you got something that looks like that. We were actually all blown away, and then the players actually got something that looked like that. It was really, truly phenomenal. We were very excited about that, particularly because it hadn't been a structure observed before in nature. And so we expected it maybe to fold, but looks like something else in nature, not something totally novel. And so we were pretty thrilled with that. But furthermore, the structure, the point was they wanted to improve function. So we set it out there because we're like, well, let's make some new interactions to improve the function of this protein. When you actually test the function of this enzyme, ignore these couple ones. This is where the protein, the very, very, very first protein started. This was my 2 years of work right here. And then this was the players. Right? And so I was like, well, I felt a little silly, but that's okay. I felt good that I gave them that opportunity. So they were about 20-fold better than I was able to do as an expert in the field through playing this video game. I'll say, I got it ready for them. So I helped a little bit. But that was fine. Oh, yeah.
How many players contributed to this?
Yeah, so there were about 1 million submissions in total, but the submissions, there's thousands of submissions for each player. So there was a few hundred players, but it really came down to about a half dozen players that continued to submit and really got the best scores. And so some players are better than others, and it kind of, there's an ebb and flow of who it is, and it's interesting. So one thing we're always asked explicitly by the players is to not reveal their identities. They don't want their username. They don't want their name. They don't want anything about them in the published articles. And we were trying to figure out why. And what I think is the reason why is if you actually look at player hours, like their usage hours, it's roughly 9 to 5 Monday through Friday with a dip at lunch. So I understand. And so that's fine with me. And so actually what we do, because we do want to give the players credit, is that we call Fold It players. When we submit papers and we publish papers, we just have Fold It players as the name, and the community gets the call. So if you search for Fold It players, you'll find citations to Fold It players. Deep hidden in like supplemental figure 12 in the online only portion, we do have for this paper the player IDs and their profession, simply because when we sent this paper out and it came back, the scientific reviewer said, well, yeah, but how do you know they weren't protein engineers that designed that structure? And we were like, well, that's a good point. We should probably figure out.
Why should you care?
Yeah, exactly. Well, if they already knew what they were doing, and it turns out none of them were protein engineers. They were administrative assistants, architects, and a lawyer, and I'm trying to think what else. I mean, it was things that they were not involved in this field. There was one immunologist, so there was a scientist. So, but I wouldn't say the immunologist is like well trained in structure of a protein. Well, maybe they are, but probably not. So we were pretty thrilled with that. We were like, well, okay, and they accepted it at that point.
Were they university administrators?
I don't know. Could explain a lot. So, it's a bad. The other joke I like about Folda is we have all these players and everyone's playing at 9 to 5, and we introduced it right around 2008, right around the crash. And so, sorry that yes, if you give people, communities, online communities, tools, it triggers their interest, and they can perform novel scientific discoveries. And so we were quite excited about that. But that's one data point, right? And so I want to show you guys another one to give you another example. And so this next one is focused on making an actual therapeutic as opposed to just proof of principle. Is this even possible, right? So we'll get into it in a second. It's about gluten intolerance. I'm assuming everyone knows what gluten intolerance is, at least that gluten exists. Yes, all right. So this next part of the project was carried out through what's called the International Genetically Engineered Machines Competition. We actually have a team here at UC Davis, which I help run, as well as Mark Facciati and Ilias Tagapoulos, and it's run from the Genome Center, with support from the Genome Center. Thank you very much. And so I've run this program since 2009. Essentially what it is, it's an international program. When I say international, there's about 60 teams in the US, there's about 60 teams spread throughout Europe, there's about 60 teams spread throughout Asia, and then there's about another 20 or 30 teams just kind of throughout the rest of the world. And so it's truly an international competition. And essentially the way the competition works is undergraduate students get together, think of some kind of project that they want to carry out, and then they carry out the project over the course of a summer, and then they go to MIT at the end of the summer and present that project. And so the way, the more detail of how we run the program is in winter we introduce them to the field of synthetic biology, which is where protein design falls into as far as overall field goes. Basically it's saying we're going to engineer synthetic biology. It sounds like kind of a weird word, but all it really means is that we're going to impart engineering principles when we tackle biological questions. So we're going to, instead of trying to understand how biology works, we're going to engineer biology to do what we want. So once they kind of have some idea, and I'm saying, winter quarter about synthetic biology, most of them are juniors or sophomores in this competition, and even freshmen. They brainstorm projects in the spring, and in the summer they carry it out. Like I said, in the fall they present and document it. So in 2011, the team that I was leading consisted of several students, but in particular, I want to point out Liz, Sydney, and Sarah, who really were very excited about the idea of gluten intolerance. In 2011, it was really starting to show up on the scene. You could actually just start getting gluten-free pizzas, and all of a sudden everyone realized that gluten intolerance is a real thing. There are people that do have it. And so they were like, well, let's do something about it. They didn't quite know what, but they knew they wanted to do something about it. Gluten intolerance is a tough problem. I joke about it, saying, it affects the staples of undergraduate life, and top ramen is not really rice noodles. They are wheat, just so you know. But yeah, undergrad life without beer is a tough life. But it is actually quite a serious problem. I have you don't know, but now you'll know, is I have a cousin that's gluten intolerant, and if there's just a hint of gluten, like a hint of he has to leave the room when you eat pizza. If there's a little bit, he has to go to the hospital. And I actually know quite a lot of people that is actually a very serious life-threatening disease. It's not, they don't like when I joke about it, but they understand as long as I give the caveat that that's fine. But it is a well-known, well-recognized problem, and it goes all the way from life-threatening to mild discomfort. They started doing some research, and what they turned out is in 2002, Jane Khosla down at Stanford figured out how gluten intolerance works. And he actually had a very vested interest because his family has gluten intolerance. And so he really wanted to figure it out. And so his lab kind of started trying to look into that. And so from wheat, rye, and barley, you have what's called gluten. That's a protein that's within there. Particularly, there's one specific protein within this mix of proteins called alpha-gliadin. And what I'm showing you here is the sequence of alpha-gliadin. And every protein has an amino acid sequence. So proteins are composed of a long, you can think of like pearls on a string, right? A long set of, with lots of different pearls, and there's 20 different pearls. So for this one of the pearls, there's a pearl for P for proline and Q for glutamine. There's a lot. There's over 50% proline and glutamine in this protein. Usually in most proteins, they're under 5% to 10%. They're very, very, very low abundance. So this is a very weird protein with all these P's and Q's, right? And in particular, so people that are gluten intolerant or gluten sensitive, you eat wheat, it goes into your gut, starts getting broken down, right? If you're sensitive to gluten, these P's and Q's and this particular stretch of P's and Q's triggers an immune response. If you're not sensitive to gluten, it just goes right through and your body never even knew it was there. It has no effect on you whatsoever. But for those that do get the triggered immune response, that sucks. So one way to stop an immune response is to maybe take a pill that would recognize this, break apart the P's and Q's in the stomach before it hits the lower intestine, and then you won't have that response because you've broken up those motifs. And you have to have those long motifs to trigger that immune system. So this seemed like a pretty simple idea. Chang Khosla already has a company, it's just hitting a second round phase 2 clinical trials that actually does this. They've taken a couple proteins that recognize this protein, so protein versus protein, and breaks them apart right at those prolines and glutamines. And they're having decent, moderate success, but they're having some real issues as far as efficacy of their drug. So the students were like, okay, well, maybe we can make a better one. And so they went through and they started to try to figure out, well, if you wanted to make a drug, if you wanted to make it from scratch, how would you do it? And they came along this protein called coumamolysin. I can't even begin to tell you where the names come from for this one. I can tell you it's from Japan. I can tell you that much. Its optimal pH for function is a pH 3 to 4, which is exactly the pH of your gut after a meal. So that's great. It's already going to work in the gut environment as far as the pH goes. But its native specificity is PR, which we were actually somewhat excited about because it has one of those two amino acids as the P, right? But it doesn't have the Q, the proline glutamine. But people said, it sort of kind of works a little bit on this proline glutamine. And so the students were like, oh, this looks actually really promising. It works at the right pH. It has almost the right function, but not quite. Yeah?
This is a native protein of some?
This is a native protein. This is a... No, yeah. So this protein is found in a bacteria that grows in hot springs. No one knows what it does.
Well,
it's there.
Right? I mean, it breaks up proteins.
It breaks up other. We know it breaks up other proteins. Yeah, we don't know like why or in the natural context why it does what it does. But yeah, it doesn't. It doesn't like to do this. So we know it doesn't treat celiac disease. But we thought maybe if we could re-engineer it, it could. And so this it goes back to where Foldic comes in. So now the students on their own came up, these are not trained professionals, they came up with their own project. They dug through the literature, had some ideas on what they wanted to do. They wanted to re-engineer this protein. But as opposed to being a 5 to 10 year process to engineer a protein and to learn how to use all the software and learn how to use all those tools, they had Folded at their disposal. They had a video game that they could open up, load that protein in, and start playing around with the structure of it. Like within minutes of finding that protein, right? Really, really, really fast. And so they went opened up the game, loaded in the protein, and I'm just showing you here is in the context of the protein, they found some mutations, and here they took what's called a glycine, where it's a smaller amino acid, into a serine. So again, concepts happening again, small to large, right? And so what I'm showing you here, you can kind of see the little blue line is a good interaction. It's a hydrogen bond between the serine and the glutamine. So with this mutation, they were able to identify a new amino acid that introduced a hydrogen bond, and their score got lower, which means it got better. I will say if you guys go and play Folded after this, because everyone can go download it, the online game version, a higher score is better, because that's what people are used to, a higher score is better. So we take the predicted energy of the system and multiply it by -1000, and that's your score. But if you play the internal version, which is no different than the external version, it's the same thing. It just gets rid of a lot of the online features of the game. we turn the score back to negative. Just because as scientists, that's more comforting to me. So we were like, okay, this is great. And so using Foldit, they designed this mutation, very similar to before. They designed a lot of mutations. And they went through, and I showed them how to do the rest. They actually built their mutations in the lab. They went to the wet lab, built their mutations, tested them, and started looking at which ones had activity and which ones didn't. So like I said, the native enzyme, the cumulolysin, had a little tiny bit of activity, but not a lot of activity. And through Foldit, they were starting able to make mutations. Some of their mutations, right in this area, were neutral. They didn't really help or hurt much. This is -1 or +1. Some of their mutations hurt a lot, so 8-fold lower. But then some of their mutations really started helping, some even over 10-fold from a single mutation. And so you can imagine they got this data set and they were like, well, that's exciting. They were able to go back into Foldit, start looking at these mutations, saying, how can we recombine these or make more mutations that look like this? And they were able to engineer a new protein where the native enzyme looked like this. And there was over a 100-fold increase in activity, and this is what their engineered protein, as far as the activity was like. And so, in the course of a month and a half, that's all this took. They went from an idea to something in hand that had a lot of activity on the exact thing that they wanted. And so then the question is, does that actually degrade and de-immunize the system? And so I will say the students ended at this point because that was as far as they were able to go. They went on to the competition, and they actually won that year the competition for this project, which was quite exciting. It's the first and only U.S. team to ever have won the iGEM competition, which is, until this year, we'll see. This year's team has a pretty cool project. If you guys want to talk about it later, I'm happy to talk about it. It has to do with olive oil, and proteins. So I want to give you one hint of the data, but I can tell you, so from the students project, there's now been a company spun out called PVP, Protein versus Protein Biologics, that's putting this protein into clinical trials, and we're about to go into, so I'm a founder of that company, and we're about to go into phase 1 clinical trials. And this is one hint of the data that's there. So we have found this protein absolutely does de-immunize gluten. If you take patient samples, their cell samples, the T cells, and you give it to de-immunized gluten, the T cells don't respond at all. Whereas if you didn't have our protein present, they light up and they go crazy. And one problem where we really thought, like I said in the beginning, there's another company that's already in phase 2 clinical trials, but they're having moderate efficacy, is that they are not particularly, the proteins that are in phase 2 clinical trials are not particularly specific. They recognize just the or just the Q, but they don't recognize PQ right there together, right? And so that, while it doesn't seem like a lot having just two things together in a specific order, that adds a huge amount of specificity when you think of the context of what other proteins are out there. So to illustrate that, in blue here is the actual, and this is the actual formulated therapeutic that's in phase 2 clinical trials, and this is not de-immunized, and this is fully de-immunized. So we want to get to 100%. Over the course of about 40 minutes, which is perfect. This is, in your gut, there's about a 2-hour resting period. So you want deimmunization to occur over an hour period. They're able to get to almost complete deimmunization, but not quite complete. So that helps sort of explain maybe why they're not having great clinical, efficacy in clinical trials. Our Coumax gets to 100%, but it takes a long time. It takes about an hour, but that's okay. You just get a little more protein, and it takes half the time. This was really just set up for this particular assay. But what I should say is this is not an actual sample. This is just water, gluten, and our enzyme. I'm assuming no one's going to eat a meal that looks like that, right? Or if you do, that's not going to be a tasty meal. So we wanted to say, okay, well, what happens if you put this into the context of actual dietary proteins? And so we went out to the store and we got what's called the protein monster from Odwalla because we figured that sounded appropriate. It should have some protein in there. And we said, well, if we just swap out the water, this has water in it, but put protein monster instead, what happens, right? So the one in phase 2 clinical trials, as soon as you put a bunch of other proteins there, it gets distracted. It starts going after all those other proteins because it's not specific. It just sees protein and says, you have a P, you have a Q, you have a P, and it kills them. But it doesn't kill the one you really, really want it to get rid of, right? And so you actually, over an hour period, you don't see any deimmunization. Not really much, anything at all happens, yeah.
Looking at the left slide, how do you have this accuracy of more than 100%?
It's not, it's just experimental error. Yeah. There's error bars on here, so. The 100% I would say is actually probably
Really effective.
Yeah, yeah. There's some error bars on here. Yeah.
If I was heading the company that is now in phase 2, I would just
They're not thrilled with
download,
us.
fold it, and
Start engineering. Well, we've patented it. All this, not folded. We've patented these proteins at this point.
but if they have like their principles similar, right?
Oh.
Like they're using a protein, they just want to be more specific. Like,
Yeah.
couldn't they just also try to use fold
They
it?
could. Yeah, they could. And so I've actually, I've talked with Chen quite a bit about this, and he's actually a great person to work with in general. He just wants to see a solution for this problem. He doesn't really care who makes the solution. And so that was actually one of the original things when we started talking to him and saying, hey, we have some students interested in this. We contact Jane and we were like, hey, what would you do? He said that. He's like, make this enzyme better and more specific. And we looked at it for a while, and it wasn't going to be really amenable to it. So I'm giving you examples of success. For every success, there's 10 failures because there's something in biology that doesn't work. So we looked at that one and we were like, well, the features of it just weren't quite right. And so that's why the students were really excited when they saw this protein, because it just looked like it had the right features, and it does have the right. Well, we think it does. It's not through clinical trials yet, but it looks very promising at this point. So what you can see is our protein's not affected. It's very specific at targets and dose. Yeah.
I just was wondering, what kind of environment are you testing these proteins in when you mix them with the new materials and the enzyme and all that?
Sorry, maybe I misunderstood. Can you repeat the question?
Where are you testing these?
Oh, test tubes. Yeah. So you have to get into people or anything past that, to go through several stages. So initially, you do everything in test tubes. In laboratories, at the university, and this is actually all at University of Washington at this point, just for IP reasons. This happened right before, literally, the company started, I think, in August, and I came in September to Davis, so it was right before. So it's all still at the University of Washington, but I talk with them daily, almost. So you start off in the lab. And then once you have leads that you really think you want to go forward with, you have to get your potential therapeutics produced in a way that's well established to make sure that you're not going to have any carryover toxins. So it's called GMP production, which is quite expensive, but it's done outsourced into specialized labs that produce protein therapeutics for people. Once you have those proteins produced under those conditions exactly, you take it and you give it to animals just to make sure it's not toxic. No one's expecting these to be toxic because it's a protein you eat, and we eat a lot of proteins. So it's unlikely. Clearly, there are proteins that are immunogenic because that's what we're breaking, but it's unlikely it's going to be immunogenic. Most proteins aren't. And so we're not expecting toxicity. And then once you have all that, you compile data, you give it to the FDA, and you ask the FDA, can we give this to healthy people? And that's phase 1 clinical trials. And so we're expecting to hit phase 1 clinical trials for this one of Q1 2017 at this point.
You said it was a protein that you eat, but I thought it was a novel protein.
Well, the therapeutic, you could imagine it'd be like the lactate pill or something like that, where it'd be you're about, you're gluten intolerant, you go out to a restaurant, you still don't probably order a pot pie or anything like that, but you can order your salad or whatever gluten-free meal you want, and you would pop this pill, and you wouldn't have to worry about, did some gluten get into it? Did they cook with flour in the back room?
But the protein in the gel itself is not.
No, it's a novel engineered protein. Yes, which was why you definitely have to go through FDA and clinical. You can't just give it to people until it's proven through. Not yet. Not quite yet. No. It's only about 2 years old, but it's going quite well.
How many people are in it?
3. Yeah. Yeah. No stock yet, though. No stock. Well, I have stock. I think it cost me nothing. It's like pennies for it. when the company, it's not really worth much right now.
Undergraduates will now be able to drink here.
I know. That's basically what it means.
You're not allowed now?
Yeah, not on my watch at least, or not in the lab, definitely. So okay, well so that's kind of where that story ends. So we have one, just proof of principle, online communities engineering proteins, undergraduates developing projects and being able to use video games to engineer therapeutics that are actually look like they're going to clinical trials. And this kind of inspired me to start trying to expand this and make it more open to society and more open to people in general. Because what was common about these two was that the inexperienced users, the ones without long term in the field, were the ones that did the simulations and did the computation, played the video game. But then they needed training to get into the lab, right? They had to have a lot of years of training to get there. And not years, in this case, I was able to help them pretty quickly because I was, a trained expert right there. But I really wanted to make this so more people could go out and do this, and it wouldn't be so hard, and you wouldn't have to have years and years and years of training to not only design the protein, but to actually go all the way through and test that protein as well, to make it so you could do something pretty fast. And so that's something we've started developing here at UC Davis, and we're calling it right now, it could change, is the standardized engineered enzyme knowledge bases, platforms that would enable people to go to your local DIY bio lab or do an internship at a university where you have the equipment and stuff like that, and carry this out, or even a garage lab. A lot of the equipment and things that we use, you actually don't need licenses to get, and you can get and do things within your, there's a lot of people that do it within their garage because the equipment's not particularly expensive. And so the idea is that people can go through, use the computation, and we've, I think, pretty well established now that they can do the design. You don't have to have years of experience to do that. But building, testing, and then learning from that and going back, I think everyone can learn. So we're taking care of that, we've taken care of that, but it's the building and testing part. How do we standardize that and make it easy for people to do? So over the last year and a half, we've really kind of spent a lot of time, and a lot of students have put into this, trying to figure out how to standardize these protocols for particular systems and make them fast, efficient, and affordable. And so we've been able to get it down to $40 in 4 weeks to go from design all the way to the learning step, which is, if you think of that, that's about, I think, 20 lattes, roughly, maybe 15. So that's pretty good. So people can actually go design it and go through all the way to the test. And so we've released this on the seek.genomecenter.ucdavis.edu. It's a very, like I say, we just released this. This is brand new, yeah.
How does selection actually occur between design and build?
The what?
To select for what is the most optimal option between designing and building?
For right now, so yeah, we have a protein up there that's relatively easy to build and relatively easy to test, and has pretty straightforward design. And well, you'll get to my, that's actually the next slide, so that's a good question. So right now we've put the first protein up there to make sure people can have everything they need to know about how to do it for this protein. Because it does sort of take an expert. I do think at this point it takes an expert and a lot of intuition and general knowledge in the field to figure out what protein you want to work on. But once you know that protein, it shouldn't be hard. It should be very accessible to people. Or if there's someone that wants to do a new protein, we can help them say, well, here's how it works for other proteins. They can template it off of ours and expand it out pretty readily. And so we're working on a glycosidase, and we've chose this protein. There's a few reasons we chose this protein. The practical reasons is there's significant interest in healthcare as far as using this protein as a therapeutic or for natural product recovery for antioxidants, as well as a lot of money that's been put into producing this protein for breaking down lignocellulose to actually produce fuels and chemicals that are important for industry. So this enzyme has a lot of interest in a lot of different fields. And so we're like, well, if we can make it so people can pretty readily engineer this, and they don't have to have years of experience, that sounds pretty amazing. And so I can tell you at this point is we've had a slew of students, and I'll point out one student right here because that's Richard's son. Came into the lab.
And thoroughly enjoyed it.
And really enjoyed it. And so they spent essentially the last year developing these methods and trying to figure out what's the most robust and straightforward way that I can give a brand new high school student that really is not experienced in this field, just give them a cookbook, like a recipe. You go to All Recipes or you go to some online site and you say, okay, well, here's the recipe on how to cook this. Most people, you can hobble through it. You can get through it. We want the same idea here. Like, how do you give someone the recipe on how to do this that anyone could follow? And so I figured the best people to make those recipes are the people that need the recipes, the people that aren't experienced. And so we've had quite a few. These are all undergraduates, except for there's a couple high school students here and a couple high school students here. But pretty much everyone else is either a freshman or a sophomore. There's one or two juniors and have not had extensive experience in this field. But they've developed these protocols. Like I said, they got down to $40 in 4 weeks to go through the entire cycle when they literally handed it to a brand new student. And at this point, they've engineered over 100 proteins for this enzyme, which is the largest. That is the largest collection of proteins that have ever been kinetically characterized. And so this group of students in the last year have done something that we haven't done in the last 200 years by enabling this rapid progressive cycle. And so we're at this point, they have one protein that's about 10-fold better than any other protein that's known for the reaction that we were looking at. But like I said, that's just the beginning here, and so I'm really excited to see where this goes. So it's kind of where I'm going to wrap up. Let's see where I'm doing on time. I think I'm like right on time. Yeah.
Can you give us any intuition on what kind of changes with lower energy in a protein molecule?
No. I wish.
Is that what you expect the user to bring to?
So it's kind of a twofold. I can tell you what I expect, but what I expect is rarely actually what ends up working.
But the program has some means.
The program does have means to calculate. The first thing I teach the students is that it's a very, very rough guide, because our ability to do computation and accurately analyze these things, it will very accurately tell you what will not work and what don't. It's not worth your time to do this, but it's really not good at telling you what will work. And so that's actually exactly the reason that my underlying academic, not for the good of humanity, but for my academic selfishness, is I want large data sets of mutated proteins with characterized activities. Because as soon as I have that, I can start picking out rules and trying to answer that question of what actually makes a protein better. And like I said, we have 100 data points now, which is more than we've ever had. The largest one I've ever heard of, I've ever seen, and I've looked for about 10 years for data sets like this, is about 15 proteins. So we have an order of magnitude more just in the last couple of months than we've ever had before. But 100 is not a lot. 100 is a very small number. We need thousands to probably tens of thousands before we can start really picking out rules. And so I can tell you that what won't work often, but it's really hard for me to tell you what will. But that's where the computation is good. It says, don't try this, and it gives you ideas on things to try. And I think that's where the human intuition really comes in, because we have this innate structural intuition about how to fit pieces together. The video games help because they can see the overall context and maybe the area we're looking in, we get something to fit really nicely, but it says, oh, you didn't look over here. And it says, you missed this piece. If you want this to look like this, you're going to have to change this over here. The computers help a lot there. Or we say, well, we know roughly we want it to be like this, but can we get it a little bit farther? And we're good at those gross movements, but the small fine-tuned movements, the computers are much better at, is what we're finding. So it's really really complementary to have human intuition with the computation. I'm going to go to my last slide. I'll finish. It looks like there's a bunch of questions, so I have 2 more slides I just want to get through really, really fast. So I did say it's ending, but it's like almost the end. So I did want to say, Foldit was the first, and at this point the most successful, probably because it was the first, and it was developed in 2008, so we're not talking a long time ago. So when I say most successful, to date, most successful, science game program out there. When I say there were community citizen science projects before Foldit, absolutely, but they were not video games. They were not driven by a score and to compete and to do better at something. They were just, can you donate your time? This was the first one where it said, this is an actual video game where you're going to get a score and you're going to be on a board ranked, and how, the better your score, the higher your ranking in the community goes. There's a lot of different programs that have now come up along this line. So if you're interested in playing games on doing DNA alignment or figuring out how to manage resources to save your forests, or analyze blood images to identify malaria, or all sorts of other things. These are all now games that are actually available and running in online. Eterna is one actually. Eterna and NanoCrafter came out of the same designers of Foldit, so there's a lot of similarities between them. And Eterna is actually, they've had some really interesting results recently where they've been publishing it. They're designing RNA structures, so nucleic acid structures. And at this point, they just published a paper, they have found that people are significantly better than computers at designing RNA structures with just very, very basic rules. The computers just can't get to where people can get to just because of the sheer number of possibilities, and people can just jump to where it needs to be. So they've had some really cool papers out on that.
How many people are playing
Several thousand, not quite as many as Foldit. I said there was several 100,000 that have played Foldit. At any one time, there's 100 to 200 people playing it, but it changes. Eterna has 20 to 30 people. I think Riju Das at Stanford is the one that runs Eterna. He would be able to give you more accurate numbers than I can on that, for sure. So with that, I'll take questions. All right.